This is the Wolbachia project. It's the preliminary survey of insects infected with Wolbachia in the Fort Towson, Oklahoma area. The entire Biology 2 class is taking part in this project. Okay, Wolbachia is a gram-negative endosymbiont and belongs to the rickettsial family. And it lives within the cells of its host.
In 1916, a rickettsial bacterium was found in the esophagus of the Culex pipiens, which is a mosquito. This bacterium was identified as Wolbachia in 1924. In 1936, it was named after S.V. Wolbach, whom was one of the identifiers. In 1950, it was discovered that Wolbachia causes cytoplasmic incompatibility. Wolbachia has both positive and negative consequences. Some of the positive consequences are it inhibits dengue fever, malaria, the Zika virus, and yellow fever. Some of the negative consequences are it causes cytoplasmic incompatibility, feminization, vertical and horizontal transmission, and it has an impact on evolution.
Okay, there are some positive effects to Wolbachia. Wolbachia does not naturally infect the Aedes aegypti mosquito, but the Aedes aegypti mosquito carries quite a few different diseases that are very harmful to humans. So what researchers are doing are gathering the Aedes aegypti mosquito and infecting them in a lab with Wolbachia. And Wolbachia is able to inhibit these diseases such as dengue fever and malaria, yellow fever, and chikungunya, and the Zika virus.
Horizontal transmission is a parasite or infection that is passed from species member to species member. In 1999, scientists thought that horizontal transmission occurred on a normal basis. In 2015, they contested this theory, saying that it did not happen on a normal basis. When they contested this theory, they found that the wasp parasitoid would lay its eggs in its paralyzed prey and in turn pick up Wolbachia in its mouth and ovipositors. When the eggs hatched, the offspring would eat the paralyzed prey and in turn pick up Wolbachia.
Vertical transmission is a parasite or infection transferred from mother to child. Vertical transmission is needed after horizontal transmission has gone through the host. Wolbachia usually undergoes vertical transmission through the maternal line of host populations.
Wolbachia is emerging as a potential evolutionary force. Feminization results in a female-by sex ratio. Feminization is when the male turns into an unfertile female. Its effect on host population can impact speciation and extinction. Speciation is the different species formed through evolution.
Cytoplasmic incompatibility is the inability for a male and a female of a species to reproduce due to Wolbachia. There are two types, unidirectional and bidirectional. In unidirectional, one insect is infected and the other is not, and they cannot reproduce. In bidirectional, each insect is infected but have different strains of Wolbachia.
There's 3 steps to the identification process. The identification is where we took the computer and the study of insects book and we look up for the order, family, genus, and species. Sometimes you can't always get down to the species, so get as close as you can. We then took the photography and she took two different types of pictures. She took one of the full body of the insect and then she used the microscope to take smaller pictures of the insect, like the tarsi, wings, and the head and antenna. Okay. And then with the drawing, she takes the identification sheets and the pictures and the insect itself, and she draws as best as she can close to the insect that she has. And once we're through with our insect, once we have all of our results in, we'll send it off to the Wolbachia headquarters.
DNA extraction. Step 1, cut the abdomen from the insect. In step 2, we place the abdomen into a centrifuge tube along with 200 microliters of lysis buffer. We then macerate the insect and add 800 more microliters of lysis buffer. We then set on cap locks and set it in a water bath at 99 degrees Celsius for 5 minutes. We then centrifuge for 5 minutes at 10,000 Gs. After this, we transfer 400 microliters of the liquid into a new centrifuge tube and add 40 microliters of 5 molar sodium chloride solution. We then ice for 5 minutes. After this, we centrifuge for 5 more minutes and then transfer 300 microliters of the liquid into a new centrifuge tube and add 200 microliters of tRNA's buffer. We then centrifuge for 1 minute and freeze until PCR.
PCR is a process that repeatedly denatures and re-anneals DNA in order to amplify it for electrophoresis. PCR is made up of 5 stages: initial denaturation, denaturation, annealing, extension, and final extension. To start, PCR goes through 1 cycle of initial denaturation at 95 degrees Celsius for 120 seconds. PCR then goes through 30 cycles of denaturation, annealing, and extension at 94 degrees Celsius for 30 seconds, 55 degrees Celsius for 45 seconds, and 72 degrees Celsius for 60 seconds. PCR then goes through 1 final stage of final extension at 72 degrees Celsius for 600 seconds. After PCR is done, we then move on to electrophoresis. First, our DNA loading dye is added into our PCR solutions, and then the gel is loaded into the tray. The DNA is loaded into the gel. The tray is placed in the chamber. The chamber is filled with LB1X fluid, and then PCR is run for approximately 30 minutes. The table above shows the results from our past several weeks of running the experiment. All insects that are written in purple are infected with Wolbachia. All insects written in green showed insect DNA and weren't infected with Wolbachia, and all insects written in black showed no DNA at all. The first column on the table, the first insect in the first column was tested at OCCC. The next several weeks were performed here in our lab. The first 2 weeks of performing it in our lab were done using a methylene blue procedure. The next week, we switched to a silver-safe green dye, which allowed us to use a UV transilluminator to better see our results and take a clearer picture.
And here our chart for the insect infection of Wolbachia. In order of Diptera, we caught 4 insects, and 3 of them did not catch Wolbachia in it, and 1 of them did. In order of Lepidoptera, only 1 insect was catch, and it was not infected with Wolbachia. In order of Platodia, we caught 4 insects, 3 was infected, and 1 was infected with Wolbachia. In order Hymenoptera, we caught 6 insects, and none of them was infected. In order Coleoptera, we caught 3 insects, 2 was not infected, and 1 was infected. In order Orthoptera, we caught 7 insects. Two of them we didn't get any result, any DNA result, and only one of them did not have any Wolbachia insect, and 4 of them was infected with Wolbachia. Here are some photo of the gel that we run electrophoresis with. In the first 2 photos we just use methylene blue, and in the last photo we use UV light illuminated. So in the 3rd picture we get much better result than the previous 2. Here's the zoom out for that photo. As you can see right here, here's all the wells, and the insect DNA, which have 709 kilobase pair, so it didn't travel as far as the Wolbachia DNA, which has 438 kilobase pair. As you can see right here, 2 lines of different. One line is insect DNA, and the other is Wolbachia DNA. Wolbachia is impacting insect evolution and propagation. The rate and the percentage of Wolbachia infection is not known because there hasn't been a lot of research made, and there's both positive and negative consequences to Wolbachia infection, and also more research on Wolbachia need to be done so that the more we will know about this.
That's a good one. All right, good. We did it! Yay!